Hello everyone, today I'll be walking you through the steps of cellular respiration, specifically focusing on how ATP is produced from a singular glucose molecule. So cellular respiration is crucial because it provides the energy needed for cells to function properly. Basically, cellular respiration is the process of converting glucose into ATP, and it involves three main stages, which are glycolysis, acetyl-CoA formation, and the citric acid cycle, followed by the electron transport chain. So the whole goal is to produce ATP, which is the energy currency of the cell. So before we dive into the details, let's talk about the significance of ATP. So ATP is the primary energy carrier in cells, and every cellular function from muscle contraction to DNA replication requires ATP. So the process of cellular respiration allows our bodies to break down glucose and efficiently use this energy. So the first step in this case is glycolysis, and it occurs in the cytoplasm, and it begins with one glucose molecule. So this stage does not require oxygen, and it can occur in both aerobic and anaerobic conditions. So one key step in this is the conversion of glucose to glucose 6 phosphate, which is G6P, and it is catalyzed by the enzyme phosphofructokinase. In the energy payoff phase, we generate 2 NADH and 4 ATP, which results in a net gain of 2 ATP. So basically, the process of glycolysis produces a net gain of 2 ATP. So once glycolysis is complete, the pyruvate molecules produced are transported into the mitochondria. So this basically shows us the two stages of glycolysis. In this case, it shows the first stage, which is the reactions 1 to 5, and the second stage, which are the reactions 6 to 10. So in the first stage, one molecule of glucose is converted to 2 glyceraldehyde 3-phosphate molecules in a series of reactions. That consumes 2 ATP. And then in the second stage of glycolysis, which are the reactions 6 to 10, like I said, the 2 glyceraldehyde 3-phosphate molecules are converted to 2 pyruvate molecules, which generates 4 ATP and 2 NADH. So whenever glycolysis is complete, the pyruvate molecules are sent to the mitochondria, where they undergo further processing. So each pyruvate molecule is converted into acetyl-CoA, which is a molecule that enters the next stage of respiration. So in this case, you can see acetyl-CoA as it enters into the citric acid cycle. So this process basically produces NADH, which can later be used to produce more ATP. So the enzyme which is responsible for this conversion of pyruvate is pyruvate dehydrogenase. And it facilitates the removal of a carbon from pyruvate, which releases carbon dioxide as a byproduct. So now that we have acetyl-CoA, like I said, it enters into the citric acid cycle, and this is still in the mitochondrial matrix, by the way. And as it enters into the citric acid cycle, it undergoes a series of reactions that further break down the acetyl-CoA to extract high energy electrons. So these electrons are then stored in NADH and FADH2. So for each acetyl-CoA molecule, the cycle generates 3 NADH, 1 FADH2, and 1 ATP. But then since we have 2 molecules of acetyl-CoA, we double these numbers. So this would mean we have 6 total NADH, 2 total FADH2, and 2 total ATP. So the enzyme that catalyzes the first step of when acetyl-CoA enters the citric acid cycle is citrate synthase. So now the final stage of cellular respiration is the electron transport chain, and it takes place in the inner mitochondrial membrane. So NADH and FADH2 donate electrons to the electron transport chain, generating ATP. So this movement of electrons drives the pumping of protons across the membrane, which creates a gradient. ATP synthase then uses this gradient to generate ATP. So as shown in the table, the total yield per glucose molecule is approximately 32 ATP. So this process is highly efficient and essential for sustaining life. So to conclude, cellular respiration is an efficient way to produce ATP, and it allows cells to extract energy from glucose efficiently. And this process basically highlights how complex and precise the biological systems are.